Chapter 4, Section 4, Protein Denaturation and Folding. Now, there's many ways that a protein can, or polypeptide can be folded into the correct shape. More often than not, hey, as it's coming out of the ribosome, as you see up here in the top left, it's just going to start its own folding. Foci angles are going to push certain regions of the polypeptide into an alpha helix formation or into a beta strand. Sometimes, that's all it takes. Protein can do its thing. Done. Other times, you're going to need some help. Some help getting that final fold in. That's what's going to be, the role of things known as chaperones. Think about it. Chaperone, when you were a kid, a chaperone is an adult that, accompanied you, aided you as you were going off to do something. They didn't do it for you. They were just there to help you. Well, that's what chaperone proteins do. They're not going to make the protein. They take the polypeptide that's almost folded correctly and they twist and tweak and get it into its final shape, its native conformation. Some of the times the misfolded protein is so far out of whack that it's going to need help. There's other chaperone proteins that are going to get it into the intermediate stage, which then those first chaperones can take the intermediate, get it to its native. Some of the times there's just too many mistakes. It's just not going to work. So that misfolded protein, even if the chaperones can't get it to fold correctly, it's going to get ubiquitinated. Certain amino acid residues, R groups, are going to get a ubiquitin added to it. Ubiquitin on a protein is a signal to enzymes in the cytoplasm to cut it up. Cut it up into peptide fragments. Those peptide fragments will then be recycled. Amino acids removed. Everything, put back into the amino acid pools. Sometimes the folding and whatnot just can't get right, but it's, instead of ubiquitinating, it's just going to be broken down. Here's the beta strand, here's the fibrils, here's this, that, or the other, which can either be utilized in other metabolic functions or, again, taken up by different vesicles free floating around in the cell and degraded that way, because in the vesicles will be digestive enzymes, the correct pH and all this to push the further unfolding and all of that denaturation of, and everything gets broken down. Now that phrase I just used, that word I just used, denaturation. Denaturation is the loss of three-dimensional structure. It's not a, yes, no. It's what degree is the loss. The greater the loss, the greater the degree of the loss of this structure, the correct structure, means the greater the degree of the loss of function. Turns out, yes, to have complete function, there has to be a specific shape. To have that specific shape, that protein has to be at a specific, pH or a specific, temperature. Any variance in that pH, you start to interfere with, ionic bonds, hydrogen bonds, van der Waals interactions, things like that. You start to lose quaternary, tertiary structure. You take it too far, you lose quaternary, you definitely lose tertiary, you start interfering with secondary. Protein unfolds, can't refold. So you look here at this figure down here, the ribonuclease, or, what is that? Apomyoglobin. Okay? Under specific temperatures, they work great. But as that temperature keeps increasing, it completely unfolds. Boom. there's this narrow band right here for like ribonuclease A. Down around here, it's some unfolding that's being made. This is just what's going on. But then as it hits this point here, 45 degrees Celsius or so, boom, shoots up. By 50, the majority of it, gone. Denatured, unfolded, no longer functioning. And you see here, with, pH, adding in certain amounts of HCl, or an HCl derivative with HCl, or however you want to say it. Denaturation, loss of three-dimensional structure. Certain proteins can still bounce back. They refer to this as renaturation. Depends on what degree you've denatured. If all you've done is interfere with quaternary structure and just a little bit of tertiary, those the hydrogen bonds, maybe some of the ion bonds, those can be reformed. But it's when you go and you denature to the point that the disulfide bonds, those covalent bonds, are broken. Oh, now that's going to be a bit of an issue. There have been certain groups, Anfinsen experiment they show here, where they can get this renaturation even if it's completely denatured and those disulfide bonds are broken. If you go, and if I remember correctly from reading about this experiment, they had to go through some extremes to get this to happen. And the microenvironments and the solutions and things they were using to get this to reform are not going to be what you find in a biological setting. So it's more of a in the lab thing as opposed to an in vitro thing. I'm sorry, it's in vitro as opposed to in vivo, in the cell, in the body. And it turns out that there is method to the madness of that intermediate folding of most polypeptides. They do it in a stepwise fashion. It's not just, boom, here's the polypeptide, and then everything kind of springs in, and it's final done. No, certain regions will start to fold around as those phi-psi angles start to twist to where they need to be. And as more of them twist and things start to happen, you start to get the right-hand helix forming, or you start to get the up-down undulation of a beta strand and things like that. The hydrophobic residues, they're twisting and getting the phi-psi angles in such a way that it pulls things around them, and so they can be removed from the presence or the interaction with water. And so this is a stepwise. It's not just a there, it's done. It takes a little bit of time. Things moving, things twisting, things folding in on themselves. And yeah, to a point, this is going to be work that's done at the molecular level by the polypeptide itself. Those R group interactions and things like that, and the phi-psi group or the phi-psi angles forcing secondary conformation. There's this neat thing they refer to as the Levinthal's paradox. Levinthal's paradox states that if you went through and you took any amino acid chain, there is an extreme number of different random conformations that could occur. But the ones that will occur and occur more often and remain there are going to be these conformations that are the most stable. Take the less energy, less change in entropy to occur. You look at it from a physics standpoint, thermodynamics. If you think of a free energy funnel, Okay. You start with an unfolded polypeptide, it folds, great, boom. Multiple pathways leading to one complete, stable, nice, smooth. It just happens. Or, let's have a nice, smooth look. It's a bunch of intermediate steps, some of them working against each other, that finally ends up with, where you want to be. Multiple stable intermediates leading to a final folded stable protein. Which one is it? Well, honestly, it's going to depend on which polypeptide you're talking about. Some of them are going to be more, A, some of them are going to be more B, and then there's going to be degrees between the two. But those chaperone proteins, you look at the figure in the textbook, or you look at the figure in the slides, your handouts. A great example of these is HSP70. HSP, heat shock protein. They got that name because they noticed this family, remember family from the previous lecture, this family of proteins became active when cells were put under stress. One of the best ways to stress, put them in wrong temperatures, heat shock. At least that's how I was told the name came from. Well, HSP70 is one of these chaperone proteins. Okay, it binds and aids and acts as a chaperone, and there are other chaperonins. These are proteins and things that aid in the folding of. But you look here, heat shock protein 70 doesn't have a complete, it's not a true globular. It's globular to that, doesn't have a set shape type of thing. It interacts with an intermediate folded, not completely, not the right way it needs to be. Goes through, uses some other chaperonins to help with, and then gets this where it needs to be. Things folded, things cut, things moved about to the way it needs to be. The figure they have here is a little misleading. Turns out the different conformations of the heat shock protein have different affinities, different, it's drawn differently to, the polypeptides. One conformation a low affinity, the other conformation a higher affinity, means want to interact more with. It's going to take these intermediate, not quite where it needs to be, and then it's going to go and help it push, pull. Not get it completely to the native state, but close enough that other chaperonins can come in and give it that last little twist and tweak and get the correct shape. Some of the times, some of the proteins you're going to need, everything's there, but it's not quite right. Great example of this is going to be protein disulfide isomerase, PDI. You have R groups over here, and here's an R group that's opposing it. These two have formed a disulfide bond incorrectly because it's supposed to be here. Isomerase will come, clip that, reform here through a bunch of chemical steps. Now, that doesn't sound like a big deal, but what if these were on an alpha helix and this is some beta strand that's here and it's distorting the overall shape, and instead it's supposed to be here. So anything that's isomerase, it's going to make the same thing, but just a slight tweak to the shape. Or you get the peptide prolyl cis trans isomerase. It goes through and cuts and pastes around proline residues, moves things around. Proline does that cyclic ring thing sometimes. Turns out we humans, or we animals, I should say, just we see it more in us humans because we live through some genetic disorders where animals, genetic disorder, they either don't make it through gestation or they're eaten while quite young. So we see some human genetic disorders that are based on incorrectly folded proteins. Amyloid fiber. It's a protein that's part of the extracellular matrix, along with collagen or whatever. It's what wraps around clumps of cells so they can be a tissue, wraps around the tissue so they can be brought together to be an organ. Well, amyloidose disease, that amyloid fiber either missing because it never folds correctly, thus the cells never process it and export it out, or what they're exporting out is completely useless because it's the wrong shape. It's not going to have a function. We see this kicks in with people who have type 2 diabetes, Alzheimer's disease, Huntington's disease, Parkinson's disease. Misfolding proteins in the extracellular matrix means that the organs are not going to be able to hold together correctly. There's going to be some issues with tissues and muscles and things like that. Alzheimer's, Huntington's, tremors and things, and the changes in the overall face and things you'll see with certain people. As we see here in this figure from the textbook, what we should get is this nice assembly of alpha helices and beta strands. But instead of everything folding correctly and giving us this amyloid peptide that can interact with others, we start to see this fibril forming, where it starts to aggregate. Instead of being individual and folding around and doing what the amyloid fibril or the amyloid protein should with other proteins, it starts to self-aggregate, starts to wrap around itself and form these clumps of protein. Clumps of protein disrupt tissue, disrupt organ, disrupt function. Hence, you start to see things, the tremors and whatnot, if it's muscular and all of that. The amyloid beta is a protein that we see associated with neurons. Not going to go into what the amyloid beta does. That peptide, maybe somebody can do it as part of your research project. Alzheimer's. We're starting to see that it's a misfolding of these proteins accumulate, starts to interfere with neuron function. Interfere with neuron function enough, the neurons start to degrade. Some of them demyelinate, some of them just die, which I know, yes, you're losing brain cells all the time, but you're generally able to maintain pathways. So you can access certain regions as needed. Memory and things like that. You know, those memory engrams are a certain pathway of neurons that when triggered tell our amygdala, hey, here's a memory. Alzheimer's, as we see here, has a tendency to interfere with. And those memory engrams are still there, you just can't access them. It's kind of scary. See the same thing, something going on, you know, Parkinson's, Huntington's, it's just where it's hitting and what it's hitting. Cystic fibrosis is one that we see a lot of in first world countries. CF, as it's referred to, there's a transmembrane protein. Transmembrane protein that is supposed to pump ions back and forth. Ions pumped back and forth helps thin out the mucus. It turns out your lungs are full of mucus right now. You don't know it because it's a thin film. Every time you breathe out, cellular escalator of your bronchioles, bronchi, pull it up into your throat, you swallow it, good to go. That mucus is to keep the alveoli from collapsing and sticking. It's also as a barrier to trap everything you breathe in, all the dust, bacterial and fungal spores and whatnot. All of that comes up to your throat. You never even know it unless you have CF. This protein, deletion of the phenylalanine, means you have an improperly folded protein, means you have a non-functioning ion pump. Means that mucus never gets thinned out. That thick mucus stays down in the alveoli, starts to fill up the alveoli. Filled alveoli have a tendency not to exchange oxygen, exchange carbon dioxide. CF patients have a lower blood oxygen levels. That mucus that's also capturing all these bacteria and fungi that's not coming back up accumulates, fills up, have a greater propensity for pneumonia, lung bacterial and fungal infections. And it turns out that this transmembrane protein also occurs in other cells throughout your body and has different roles than just the one I talked about with the alveoli and your lung epithelium. And the scariest one of them all, prion disease. Prion disease, mad cow or Khufu, if you're a Pacific Islander and you still do the whole cannibalism thing. Khufu, Kuru, I can't remember if it's Khufu or Kuru. I have to look that up. Sorry if I'm pronouncing it wrong. I believe it's Kuru. You get it by ingesting prion-infected neuron, brain tissue. Prion protein, PRP. PRPC, as you see right here, is the normal shaped protein that you're going to find in neurons in mammals. Yeah, the protein's already in your body. Protein's in your brain. Protein has a function. What's the function? Don't worry. Don't know. Don't care. Just there. Okay, it has a job. It has a function. Along comes PRP-SE, scabies. I believe is the SC stands for. This PRP-SC will interact with this, as you see here, and it forces a misfolding. Takes PRPC, forces it to misfold to become PRP-SC. Creutzfeldt-Jakob disease, Kuru, two forms that we've seen that have been characterized in the medical journals. Kuru is more prevalent, Creutzfeldt-Jakob disease not so much. So what this means, whether you're a cow or a human, you have to eat brains that already have PRP-SC in them. PRP-SC does not get broken down by the digestive enzymes or any of the proteases in the stomach. It's absorbed. Can't remember if it's absorbed in the stomach or the small intestine. It's absorbed as is, traffic through the bloodstream, settles out in the brain. It's able to cross the blood-brain barrier. Again, how? We have no idea. Gets into the brain, gets into the neurons, does this. You start to accumulate these aggregates of PRP-SC. These aggregates start to interfere with normal neuron metabolism. Neurons start to die. As you see right here. As the neurons die, they leave little blank spaces, little bubbles within the tissue, the brain tissue. This is where they get the spongiform encephalopathy. Encephalopathy means infection of the brain tissue. Spongiform, well, the first people that characterized this, they were cutting up the cow and the human brains, different times, different people. It reminded them of the sponge you have at home over your sink that you use to clean dishes with. Had that sponge, all those pockets of nothing. The cells that were there died, liquefied, and were taken up by neighboring, thus passing on more of this PRP-SC. Side note, just kind of a thing to end on. You can quit now or you can listen to this cool story. Notice a few minutes ago I said you have to eat the brain tissue. As an undergrad, back in college, worked on the dairy farms at the school I was at, and things like that, and talked to people, and I followed along with it. They're what they do to feed the cows, and for the most part, they feed the cows, plants, plant material, herbivores. Turns out cows are more omnivore than we would think about. Cows are cannibalistic. Turns out some researchers, and I forget at which ag school, at which university, took feed that was all plant-based, feed that had cow parts. You slaughter a cow. You can use the skin, the muscle, the meat, and all that stuff. Waste products, certain bones, tissue, bones, and the spinal column, brain, waste. Somebody took that, ground it up, because it's calcium, the bones, and lots of fat, because remember, your brain's mostly fat. And they mixed that up, put that in with the feed, and basically set both of them out and see which ones the cow preferred. Turns out the cows prefer the taste of cow brain. So go think about that.